Just an outline of what I'm going to talk about today. Just quickly, a working definition, where microbes exist in the body. So, and then how can we engineer the microbiome as a therapeutic? So, just a show of hands, how many people, when they saw this talk title, thought I was going to talk about putting a circuit into a bacteria and then feeding it to a person? Small amount, didn't think about it at all. Well, I'm actually not going to be talking about that at all today. I'm mainly going to be showing case studies on using the existing bacterial diversity for direct use as a therapeutic. And so the areas I'm going to cover are cancer, drug discovery, and case study for MRSA, diabetes, and diseases of the gut, and then probiotics as a diet. All right, so there's been a lot of research and investment into the microbiome, and this actually dovetails nicely with the genomic stuff. As the genome, the cost to do sequencing lowers quite a bit. People have started to think about large-scale genome mining projects, and so there's the Human Microbiome Project, where they've taken 5 healthy human subjects and swabbed a majority of their body sites and used deep sequencing, looking at their 16S RNA, and they do metagenomic shotgun sequencing to uncover the human microbiome. And just as a working definition, the microbiome includes microorganisms, but not just limited to bacteria. It also includes fungi, viruses, phages, etc. Okay, and so from this site, this is a graph of all the sample diversity for a given site. And not surprisingly, there's a lot in your feces and kind of the areas of your body that are directly in contact with the outside world, including your gut, vagina, skin, etc., etc. But surprisingly, in recent times, there's been new evidence that previously sterile areas of the body actually have a microbiome. So there's a paper that the placenta actually harbors a unique microbiome and might be involved in fetal development. And then there's evidence that your bladder actually isn't a sterile environment either, though that's a little controversial because people think that it might just be contamination due to sequencing. Because to note, for all the sequencing, you're not actually culturing the bacteria. And so that's a drawback, but also the power of the technique, so you don't have bias in your samples. And so, I know you can't read this text, but this is just a chart of all the essential chemistry that different bacteria can carry out within your body, specifically the gut. So different species make short-chain fatty acids, bile acids, choline metabolites, phenolics, indoles, etc., vitamins. And so I think one of the really interesting questions out there is what percentage of your daily intake is generated by your actual microbiome converting sugars into these products versus what you actually intake with, directly through your diet. Right. So this is the bottom-up engineering part, which I just wanted to throw a shout out to. The top panel is what we as synthetic biologists would think of as manipulating the microbiome for therapy. So you have a synthetic circuit that you put into a disease state that can sense and solve a deficiency. The top right panel is an example of how you use quorum sensing to block a virulence pathway in bacteria. And then there's been a lot of work using phage-enhanced killing to increase the efficacy of antibiotics. And so I just put a few labs that are working in this area. There's obviously many, many more, but I'll just be talking about the case studies of your extant microbiome as a potential therapy. All right, so I'll start off with cancer. So from this blog is one of the first kind of major popular science things that I read that really got me interested in this area and made me think of the broad potential as a therapeutic. But it's long been known that your microbiome actually has some bad characteristics. So like the human papillomavirus causes cervical cancer. There's different viruses that cause lymphoma. Heliobacter can cause gastric ulcers and cancers. There's new evidence that bacteria can enter your bloodstream or your gut and cause colon cancer, etc. And then case 3 is just evidence that different cancer drugs can actually be. So case 3 is where the liver inactivates the drug, but then your gut bacteria reactivate it and causes chemotherapy-related diarrhea. And so there's interest in creating inhibitors for these gluconeoredases that could limit diarrhea side effects. And then there's also tumor-associated microbes that can inactivate different analogs that are used against cancers. And so obviously, if you could do target a specific species like mycoplasma, then you can reestablish the efficacy of the drug. And recent studies have shown that the diversity in the gut can influence whether a patient will benefit from immunotherapy or chemotherapy. And that's what I'll show really quickly here. So these 3 articles all came out in, I believe, Science within the last couple months, all surprisingly on the same problem, basically showing that your immune response can be modulated actually by your gut bacteria and the metabolism that they carry out. And so they're able to map outcomes to the different classes of bacteria. So on the top right, they're showing progression-free disease, where the high abundance of this type of bacteria that's actually good shows much higher rate of progression-free disease, whereas if you have a low abundance, it's not the case. And then they actually map this causative bacteria, where if you have it, you have a much faster disease progression. And so this, to me, seems like it'd be a great treatment opportunity, just very quickly in the near term, developing a probiotic that you could give to patients along with chemotherapy to prevent treatment-resistant cancers. And then also in the studies, they showed that treating with antibiotics before starting chemotherapy can lower the diversity in your gut and also limit the efficacy of the treatment. Right. So I'll just quickly show another few figures from a paper on using the diversity of the microbiome to find a drug against MRSA. So this was a Nature Chemical Biology paper where they did a bioinformatic pipeline analysis to do structural predictions of these non-ribosomal peptides. They then actually synthesized those and then tested them against MRSA. And so here is just the structure. You can see it's a pretty complex molecule, but they're able to chemically synthesize it, and then they tested it against a panel of different bacteria that are common in the gut, and they found that the minimum inhibitory concentration was higher against these different staph bacteria. And so when you treat with antibiotic and this new huminomycin, it greatly enhances the effectiveness of the treatment compared to just the antibiotic or just the huminomycin in a mouse model. And the next area I'll talk about briefly is the diabetes drug metformin, which is commonly given to pre-diabetic or type 2 diabetes patients to modulate their blood glucose levels. So this is another very recent study that shows that the gut microbiome is actually altered by metformin, and metformin can enhance the growth of specifically beneficial bacteria. And so the panels here are showing control versus metformin growth of 2 different helpful bacteria and as a control. And then when they actually fed germ-free mice and let them colonize, they found that they're able to these bacteria are able to colonize the gut and keep the blood serum glucose levels lower than in the control model in the glucose fasting test. And third to last, I'll talk about diseases of the gut. So there's a lot of evidence that your microbiome can enhance or prevent the effect of inflammatory bowel diseases like IBS. And so, just to note, most of the stuff is in the mouse model, so you can argue how well this translates, but it's interesting information. So what this panel is showing is in a mouse model where they give these wild type OVMs, which have this capsulated polysaccharide A, they have an enhanced immune response. This is IL-10 production of T cells, and this matches to the known response due to this PSA protein. And kind of counterintuitively, this immune response can suppress mucosal inflammation in culture. And so they had a panel of experts do pathology on the colons of mice. And so in the sham treatment, nothing happens. When they have this DMDS colitis model, which is ulcerative colitis, they found that if you treat with these OMBs, you get a much lower colitis score versus treating with PBS or OMBs in this protein knockout. So they're able to show that your gut bacteria, in combination with this protein variant, can modulate the effects of IBS. All right. My personal favorite topic of this is poop pills, aka fecal transplants. So this has gotten a lot of popular press lately. And so people have been basically harvesting feces from healthy individuals and using it to treat recurrent C. diff infections, and then give the patient an enema or packaging in pill form. And they've seen, this sounds made up, but they've seen 90% complete cure rates using this treatment. And I'll show a randomized trial actually showed this had higher efficacy than the current standard. So this case study was published in the New England Journal of Medicine, and it actually worked so well that they had to stop the trial early because it was immoral to continue not treating patients. And so here's a graph, like 94% cured without relapse versus vancomycin, which is like the last defense antibiotic. And I'm not exactly sure what bowel lavage is, but yeah, basically really, really effective. And so, not surprisingly, there's been a rise in do-it-yourself fecal transplants. So I was able to just Google this. They give you a shopping list. All you need is saline and an enema bag. It's shown efficacy for IBS and also ulcerative colitis, and they recommend ground piles. So I thought that was kind of fun. It's a very nice take. Right. So this isn't the problem with this method is it's not scalable. So to get donors, only 3% of donors pass the really stringent screening protocols, and to get a sample, you literally when you move to the bathroom, you go in, provide your sample, and go back. So you can't just have humans hooked up in a lab with like feeding tubes producing this. So there are a lot of biotechs working on making culturable strains for high quality scale up. And then eventually you would imagine that you want to find the actual effective molecule and just deliver that if it is a therapy. But this company actually talked on campus a couple of years ago. That was really cool. They have their C. diff treatment in phase 3, and then here these are their synthetically fermented, where there's no need for a patient sample, and they tightly control what's in it. And so they're looking at IBS and other, like the PDL1, which I just described. All right, and so the final little thing I'll talk about is based on all this evidence of how the microbiome can be used to treat a variety of diseases, I imagine that it definitely could be a new antidote. And so this study was actually, again, in a mouse model, but they gave mice different diets and then had them diet, fast, etc., and followed their microbiome over time. And they're able to use machine learning models to predict the signatures that would show resistance to weight regain after like a dietary intervention. And so it's not a far stretch to imagine that this also occurs in humans and that you could uncover the species needed to develop an effective kind of weight loss, or probably more appropriately named homeostasis pill for people struggling to lose weight. So in summary, the human microbiome is still being defined and discovered, and it's a source of essential biochemistry. It can be a force for bad, but also good, as it's a known cause of cancer, exacerbate side effects, but it can also be a huge area to develop therapeutics. And so that's all I have, and thanks for your attention.
Yeah, so I'm just curious. I know it wasn't the primary thing you tried, but it seems like the sort of using the native microbiome is further along in the ideas of synthetic biology interventions. Like, are there any clinical trials happening in synthetic biology interventions, or like any support on that side?
I doubt it. And so the really cool thing about this and like the series, they basically got approved as there's like no side effects from like the fecal transplant stuff. So they have like a streamlined application process with the FDA where their therapeutics are generally recognized as safe. And so with like a symbiotic approach, you have to do like recombinant DNA, different genes, containment issues, etc. And so I just think that this is probably, at least in the immediate future, a way to go.
I'm going to try to squeeze in the newsreel, I guess. Thank you so much. If we get kicked out, then run. I don't know.
Thousands of years ago or something.
You think so?
It seems like it's such an easy thing to do.
But you'd have to like have some sort of evidence that you'd have to have made some observation in nature that would have some sort of effect. I think nobody would have had that idea without.
Cool. All right. I'm going to go through the newsreel of the month rather quickly, but I want to start off by pointing out the most important news of the month, which is that there's a DNA emoji now. And it has been fixed, so originally it's left-handed. Look at that. They did fix it. Apparently this came out at the same time as a lobster emoji that had 6 legs instead of 8, so like, whoever's making these needs to get their act together. For some reason, all the news I chose this month was just like depressing, so I'm sorry about that. February was not a great month for biohacking, which kind of started out with this event that happened at a body hacking convention in Austin. This is Aaron Draywick, who's the CEO of a company called Ascendance Biomedical. These are the same guys that kind of got some controversy late last year when someone injected himself with an unprecedented HIV treatment. This time Draywick got up on stage and injected himself with a herpes treatment that would potentially be a vaccine that hadn't really been validated before. There was one paper and it had only been done in mice. It got a lot of attention in the press, and then it kind of just sort of spiraled out of control. So some news reporters went to Ascendance's sort of community biohacking space, and the same day, a lot of these people in Ascendance were sort of rebelling against Draywick because they thought that he was just sort of doing this for the publicity rather than the science. So then he locked himself in the lab and like changed all the key card access and no one could get to him. The reporters were all there. It was really sketchy. And there's like some really sketchy science things going on here. I think it's important to point out that like this sort of thing like shines really badly on the whole biohacking community as a whole, and I think it's really terrible press. Josiah Zayner, who in some ways has sort of inspired this whole idea with the injecting himself with CRISPR a few months back. Like there's a really candid interview in The Atlantic saying that he feels bad about sort of inspiring these people that have a lot less scientific knowledge than what he was doing, which was sort of for a different purpose. And he has sort of a Facebook post where he's calling out a lot of these people out of science for doing this the wrong way. It'll be interesting to see how we move forward with this. The FDA has kind of recently been taking a stand against these sort of do-it-yourself treatments. It'd be good to see what happens in the future. I took out the bit about George Church because Mention did a much better job than I could explain about the genomic stuff there. So we'll move on straight into making horsepox. So this is a lab from Canada. I think it's Calgary. I'm not entirely certain. David Evans synthesized a horsepox virus vaccine from DNA fragments. Literally just ordered 10 fragments and stuck them together. This is a problem because horsepox is a pox virus like smallpox. It's actually extinct. There's one copy of this virus that exists in the world. It's at the CDC. Evans says that he was doing it because they wanted to make a vaccine that would be useful against smallpox, but he never really asked the CDC for their version of it, and it's really sketchy because, like, how did you make this vaccine? It apparently cost less than $100,000 to do the whole thing. He claims it didn't require that much expert treatment or expert knowledge at all, and that anyone could do this if they just know how to do a Gibson assembly. Science got a hold of this, of course, and had a field day. This actually happened a few months ago, but didn't get published until this month. And he tried to submit it to Nature and Science. They both rejected him, probably understandably. So it wound up in PLOS. So then Science writes an editorial like, "This is horrifying. Don't do this, people." One of the reasons they got it actually is because Drew Endy, for some reason, was serving on like this pox virus committee thing in Europe where this news broke out, and Drew Endy's like, "Wait, this is a bad thing. People should know that this is happening." And then it like reached the mainstream media. It got onto NPR. People were saying if this guy was really just trying to do research, or was it for potential profits? Because he's sort of got a company spun out of this now. So pox viruses, not good. Don't do it.
Those sequences really show up in red flags at the synthesis company?
I think it's partly because it's it's an extinct virus that nobody really knows much about. I guess it's not similar enough. I don't know. It's a little bit scary. Yeah. This happened at the end of January, but the Wall Street Journal reports that since 2015, 86 people have been CRISPR'd in China. Yes. So I think we kind of knew that this was happening behind the scenes, and there have been, some small stories that have been out there about edited. I think one person had lung cancer that was treated by CRISPR edited cells, but it turns out it's a lot wider of an issue than we had planned on. So there's all of these different clinical trials that have happened with CRISPR in China. By contrast, the first clinical trials in the U.S. are scheduled to happen later this year to treat sickle cell. This is just kind of has to do with the medical system in China too, so they don't really have the equivalent of an FDA. They have like sort of an IRB would be. They have to get ethical approval to do things, but not necessarily scientific approval to do things. So there have been, I think, 15 of them died. The doctors say that it's because they had cancer already, but it really sort of remains to be seen. Lori Zoloff actually gets interviewed in this article, so if you're familiar with some of the work that she's done, it's an interesting story for sure. And then finally for some hacker news, I did not know about this. This is really exciting. So iGen is great. It really gets undergrads and high school students involved in synthetic biology, but a lot of that research doesn't really wind up published. It sort of gets abandoned to the void. So PLOS, this is the second year that they've done this, is essentially teaming up with iGen to try to come up with a way that we can get this research out to the public. So iGen can sort of prepare research articles, and then they can post them online, and grad students like you can go on and give feedback and support and ideas for how this could actually wind up into real peer-reviewed published format. This is happening today. This is the day that it's doing it, so I recommend if you've got 20 minutes this afternoon, go log on, see what it's about. It might be really interesting. I looked at a couple of these, I was very impressed by what they managed to come up with in terms of, I would believe that this is a real paper, even if it's something about Mars missions. But hey, apparently you can buy paper and Mars, so there you go. Into the research. So Nature Chem Bio has a really cool paper on a novel way of doing CAR T therapy. So CAR T cells are T cells that are engineered to respond to surface antigens on tumors and then activate a genetic program to kill the tumor cells. Well, wouldn't it be nice if you could do it not just to an antigen on the tumor, but also a secret soluble factor? So these guys engineered CAR T cells that respond to first GFP, because GFP, and then also like factors that you might find around a tumor, cytokines like TGF-beta. And it works really well. They can get efficient killing. And a couple people in the Leonard lab, this killer here in the back, Patrick, they wrote a really neat opinion piece in Nature Chem Bio about this, so definitely worth checking out that. This last week had a really awesome issue of Science that had some really cool synthetic biology things in, and one of the highlights was Sherlock version 2. Sherlock is a collaboration between the College Lab and Feng Zhang. They're using these Cas12 proteins, which sort of act like Cas9. They can detect a nucleic acid target using like a standard guide RNA type approach. But if you think of Cas9 as a pair of scissors, Cas12 is sort of like a nuclear bomb. As soon as it detects its target, it's just like, I'm going to destroy everything. So they put a bunch of fluorophores next to it and destroys the fluorophores, and then you can use it as a diagnostic platform. So that was Sherlock version 1, which was absurd in its own right. And then Sherlock version 2 just takes everything to like completely illogical extremes. So they now can detect multiple viruses at once using different like versions of these Cas12 proteins from different organisms. It's quantitative, it's colorimetric, they increase the sensitivity, so now they get it down to 8 zeptomolar, which I'm not convinced is like real. That's 8 times 10 to the negative 21 molar, or roughly 1 RNA in 500 microliters. So like, I don't think that's real, but then they go on in this science paper that has 3 figures and 35 supplemental figures. They go on and show that you can use the Cas12 simultaneously to detect a single nucleotide polymorphism and then also repair the single nucleotide polymorphism, because this is the same Cas protein that they use for like the RNA guided repair. So they just like cure cancer at the same time. So yay them. Jennifer Dowden on the same article has sort of her own version of this, which I couldn't detect much different than this, other than the fact that the acronym is way better. It's Detector DNA Endonuclease Targeting CRISPR Transporter, which is way better than whatever the hell Sherlock stands for. Just saying. Heard it here first. In the same issue of Science, right off the bat there, David Liu's lab comes up with a really neat way of recording cellular events in DNA. So this is something that sort of puzzled people for a long time. People have tried strategies like recombinases, but those really only can sort of digital information, whether or not the DNA is flipped in or out. This is a way of being able to detect events. They actually come up with 2 strategies of doing this called Camera 1 and Camera 2, which is also a really silly acronym. They're both based on CRISPR, so one of them is based on guide RNA, mediate cleavage of one particular sequence versus the other, and then you can measure the plasmid copy number variations. And then the other one actually involves this editing mechanism that I was talking about earlier. And they adapted it to work for a bunch of different stimuli, so proteins, small molecules, light, whatever. Works in bacteria, works in mammalian cells. It's just super cool. This is a follow-up kind of for some stuff that happened last month. David Baker showed that you can design de novo protein capsids that encapsulate their own DNA. These guys were like, yeah, but let's make them out of antimicrobial peptides. So they do. And they go through all the design rules of how you can actually get nicosahedrin made from these things, and then they just like kill bacteria really well. So if you just incubate these things with bacteria, the bacteria will die in less than an hour, which is really neat. And then I'm going to go through some quick hits with papers that I'm going through in even less detail, which means I read them in even less detail. PNAS has a paper from the Church Lab. This is taking their Amaryllis strain that they developed and basically subjecting it to rounds and rounds and rounds of laboratory evolution to determine what are beneficial mutations to improve its fitness. So they report on that and some interesting mutations that developed. Also in PNAS, this is a paper about designing E. coli as metabolite biosensors. So metabolite biosensors wind up being transcription factors that regulate the expression of life